I'd like to welcome everyone and thanks everyone for joining us for the ARI seminar about saving our iconic Banksias. For those who don't know me, my name's James Todd. I'm the Acting Chief Biodiversity Officer and Director at Arthur Royal Institute, and I'll be chairing this seminar. And I've got technical support from Andy and Tess operating in the background. So I'd like to begin by acknowledging the traditional owners on the land which we're variously meeting. Pay my respects to elders past and present. I'm on the land of the Wurundjeri at the moment, and pay my respects to them, and also to traditional owners on any of the lands where you might be coming in from today, and to any traditional owners or Aboriginal people who might be joining us. So today's seminar is really taking a look at the science to be used to inform management and further conservation actions to protect our iconic Banksias in Victoria. We'll hear from ARI's Annette Muir discussing suitable fire regimes for hairpin Banksia persistence, and also Laura Simmons from Royal Botanic Gardens, who will discuss the conservation genetics and actions underway for some Banksia species post the Black Summer bushfires. Given we have started a bit late, we might go a little bit longer if people have got time, but we'll try and finish it up by 2 o'clock as per the normal time. There'll be time for questions at the end. And if for the people who are online, if you want to include your questions in the chat, then Tess and Andy will work behind the scenes and we'll come back to the ones which perhaps are the most relevant or are getting the most thumbs up from people online. So without further ado, I'd like to welcome Annette and Laura to today's seminar, and starting off with Annette. So over to you, Annette. Thank you.
Hello, thanks, James. So my talk today is about ARI's research over the past decade into hairpin Banksia in Victoria. So our knowledge about its demography is helping focus our management efforts, but it also raises concerns about its persistence into the future. Our findings have relevance for other widespread ecologically important species that are vulnerable to decline as the climate changes. Before I go on, I just want to acknowledge that this work has succeeded because of the funding and support from the Gippsland and Port Phillip regions of DECA over many years. Firstly, I'll start with a bit about hairpin Banksia and why we've been studying it. So it plays an important ecological role, providing nectar for birds and small mammals. And I just want to touch briefly on taxonomy because this has consequences for the conservation decisions about our Victorian populations. So Victoria's hairpin Banksia, Banksia cunninghamii, is part of the Banksia spinulosa complex. You can see the map on the screen. It shows the broad distribution of this complex, with the southern cluster of Banksia cunninghamii, the one we're interested in, which is represented by those blue squares in the red circle between the letters M and STZ. So this map comes from a paper by Wilson et al. That found that this cluster's distinctive morphologically and genetically, and the cunninghamii that we have in Victoria is distinguished by not having a lignituber, and I'll explain later why this has significance. As a general sort of overview of what's facing this species and other widespread woody species, is that obviously it's climate change. There are many studies showing the connections between climate change, more frequent, severe and widespread fires, and of course we remember the widespread and terrible 2019-2020 bushfires in southeastern Australia, which you can see the extent of in that map on the screen. And with these changing fire regimes, these are sometimes not suitable for species like hairpin Banksia, and there's predictions of their decline. And there's a number of articles that point to examples of other species where this is the case. So Hairpin Banksia in Victoria, it's an obligate seeder. That means that it's killed by fire and doesn't resprout. And then the seed is contained in the woody cones that we're all familiar with, which open after fire, but there's no seed stored in the soil. And then after germination, the plants need to grow for enough time to produce seed before the next fire comes around. All these attributes mean that it's vulnerable to decline with short fire intervals because of that time it takes to grow and produce seed. But we still don't know very much about what role fire severity plays in its life cycle. So we started this work quite some years ago because we wanted to know the fire interval needed to reach reproductive maturity for hairpin banksia, so that for one reason, for planned burning. So how often could burning be carried out and still retain the populations? So we started in 2014, 2015, 2016. We started monitoring cone production on Banksia plants Banksias, hairpin Banksia plants in East Gippsland and east of Melbourne. We had sites, we were focused on fire intervals, so we had sites that had time since fire from 5 to 35 years, and we measured the number of cones on adult plants. You can see this graph, and this was work that Paul Maloney did then. It shows time since fire on the x-axis and the number of closed cones per plant on the y-axis. So there's not very many, as you can see. But the thing to see there is that the line, both the red and the purple lines, represent the mean number of cones per plant, with a 95% credible interval shaded either side. So you can see in the first 8 or so years after fire, there was little seed production, and then by 15 years after fire, the maximum amount of cone production had been reached. So you see we had gaps in our data, but we were getting an idea of the length of time between fires that was needed in terms of seed production. The next question, of course, we didn't know if recruitment, that's seedlings coming up, was replacing the adult plants killed by fire, and what were the effects of fire interval and severity on the population persistence. After the 2019 Bunyip bushfire and the 2019-2020 East Gippsland bushfires, we went back and measured hairpin Banksia seedlings in these burnt areas. These were largely in lowland forest, EVC, so we focused on that where they most commonly grew, and we had sites that were our monitoring sites plus some extra ones from the VBA. So once again, we had time since previous fire, a range from where we thought they were too young to have cones, 5 years, right through to 37 years, so quite old plants. And we had a range of different fire severity experienced at our different sites. So there was either quite moderate sort of severity where the eucalypt canopy was scorched, that's where the leaves are browned off, right through to where the eucalypt canopy in those forests was burnt, so completely blackened, and the loss of the tops of the trees. And we set up plots around Banksia, the adult Banksias, and counted the number of seedlings. Essentially, that's what we did. So these are the results on the seedling recruitment. So once again, Paul Maloney did this work. This is just the East Gippsland data set of 25 sites. So the relationship between seedlings being present or absent at a site, fire severity, and the fire interval threshold was modelled, and this showed there was a clear difference in the probability that a site would support recruitment based on the fire severity in those 2020 bushfires or the fire interval preceding those fires. So you can see by that, the graph on the screen, that regardless of the fire interval, if the fire severity was high, then there was a very low probability of seedlings being present. But at sites with moderate fire severity, the fire interval still needed to be greater than about 12 years for the recruitment to be probable. So that pointed to the fact that severity was playing high severity was playing a role as well as interval. That photo on the screen just shows one of the Banksias we'd been monitoring over the years, and that had been burnt at high severity. That blob on the stump of the burnt Banksia is one of our metal tags that was melted, and there were just stumpy stems left on the plant. So I think I just want to point to the fact that the new finding from our study was that high severity fires, these very high severities that were enough to burn the tree canopies, they corresponded with almost complete recruitment failure for the hairpin Banksia. And once again, it was, when we looked at that on ground, we could see no evidence of any cones, just really stumps of the old plants, if that. So there are not very many studies on the effects of high severity. One recent one is by Whelan and Eyre, that's up on the screen there, looking at Banksia spinulosa in New South Wales after the same bushfires. Generally, studies have shown over the years with Banksias that relatively high fire intensity is needed to open Banksia cones to release the seeds, and then the seedlings can germinate. And very low intensity fires sometimes have been related to the cones not opening as much. But there's a lot of work to be done in that area, and these have been done with other species, so we're really not sure with our species here in Victoria until this recent work we've been doing. So we got that piece of information. So our next question is, or was, what's the likely population persistence of hairpin Banksia across East Gippsland, given its demography, what we knew about its life cycle, and the spatial distribution from fire mapping that we had of short fire intervals and high fire severity. This work was done by Nev Amos, and the way he did this very briefly was that the habitat distribution model for hairpin Banksia in East Gippsland, so that's the likely areas or the suitable habitat for the species, not necessarily where it doesn't occur everywhere in those areas, but that's its possible habitat. This was overlaid with the mapped fire intervals going back to 1960, and also areas burnt at high severity going back to 1998. This is where the mapping was more reliable. So using the findings from Paul's modelling, Nev then was able to see where there were areas that had been burnt either above or below that 12-year threshold fire interval, and where there had been high severity fires. So I know there's two maps on the screen there. Perhaps look at the bottom one. The blue, just to look at that, represents those areas that had been burnt at greater than 12-year intervals. So we would expect that hairpin Banksia would persist in those areas over the years. All the other colours, not black, but the other colours represent different times where there had been shorter than the 12-year interval. So we suspect that in those areas there wouldn't be enough seed. And the black is areas burnt at very high severity where we're concerned that perhaps there wouldn't have been recruitment either. So there is a caveat to this, of course, with this modelling, that burns will always have been more patchy than the mapping shows. So there's likely to be and to be Banksias occurring in those areas that we predict there'd be none. So this is a further bit of work that needs to be done. But in general, what does this mean for the persistence trajectory of hairpin Banksia? So looking at that mapping that Nev did, the modelled map, that showed that 67% of its distribution were burnt at these intervals of severity that we think is below the threshold for seedling recruitment, bearing in mind the caveat about the patchy burns and the incompleteness of the mapping. Regardless of that, though, there is a future risk that even the ones that did germinate after this last bushfire, 80% of the populations in East Gippsland were burnt and are now immature. And we know very clearly from their life cycle that it takes about 10, 12 years before they have enough seed to recruit. So if there's another fire before 2032, we're looking at some local extinctions where they're just too young to have seed and to reproduce. So this leads me to just point out that based on this alone, it does meet some of the IUCN Red List criteria of being threatened species. It's not formally listed at the moment, but I think that's certainly another step, because there has been an observed reduction in its area of occupancy in the past and also predicted into the future, and those threats are continuing. So, but I want to then just move on at the very end of my talk to say, well, we know this information. What are we doing with it, and what can we do with that information to help protect and protect the populations of hairpin banksia into the future? So already our strong collaboration with regional fire planners and biodiversity staff, we've been able to work very closely to design this monitoring so that it answers questions that are directly relevant to their fire planning. And some of those findings have been incorporated into strategic and operational fire planning. And I'll give you a couple of examples. In Gippsland, for example, they have a planning unit prioritisation tool. They call it traffic light tool, I think, colloquially. Anyway, that they as a precautionary measure are trying not to burn hairpin banksia at less than 14-year intervals. And in Port Phillip region, there's been some recent on-ground mapping of where the actual hairpin banksia occurs in some of the parks, rather than this sort of just point locations or habitat models. So this is really where it occurs on ground, so it can be incorporated into fire planning. Still, there are big challenges, obviously, in protecting these remaining populations while managing the fuel risk. We need to think about fire intervals so that the plants can produce seed and the seed's there before they're burnt. Suitable weather for recruitment, so it's not too dry so that seedlings can grow. Excluding some patches where they're too young, and certainly some ongoing monitoring. Obviously, fire planning is only one part of the thinking about how to conserve the hairpin banksia. We can't manage all these risks just through that. So there are other approaches or additional approaches that are complementary to try and prevent further decline. I think I've mentioned, obviously, listing would raise its profile for conservation actions. There's some work on its taxonomic status so that we can be clear which parts of its spread in that broader spinulosa complex are being conserved, and Laura will talk about that more, of course. Seed banking and restoration are probably going to be part of the mix where populations have really gone downhill. There's some great examples, inspiring examples of some seed orchards set up for silver banksia and other Banksia marginata in Victoria by Friends of the Forgotten Woodlands and Euroa Arboretum. And there's a little bit of work in the Dandenongs I've been involved with other volunteers, re-establishing some insurance populations, and we'll hear more about the seed banking restoration aspects from Laura as well. I just want to finish up now with once again acknowledging that the support and funding from Gippsland and Port Phillip regions. It's been very much a joint effort, and there's been so many people involved in this over the years. I just am very, very grateful, and there are just a few names on the screen there who've been particularly involved in recent years with the Banksia recruitment studies. And I just want to wrap up with my key messages, of course, to take home. So there is a trajectory of decline for hairpin banksia in Victoria, and this is based on what we know about its demography, its life cycle, its seed production, seedling recruitment, and the spatial mapping. So we know from the results that they need to be protected from fire for greater than 12 years, and very severe fires are a problem as well. And finally, it's really through collaboration that we'll get results with this, that we can, in particular in DECA, and use these findings to inform fire management. So the last thing I want to say is check out the Banksia page on ARI's website, and I think Tess will provide some more links. And that's it for me, and thank you very much for listening.
That's great, Annette. Thank you. Can you hear me okay?
Yes, thank you.
Great. I was a bit robotic before, that's why I had to come off and back in. So well done. We might hold over questions to the end after Laura's presentation, which is obviously there's some synergies between the two, but there's a couple of questions in the chat there already, Annette, you might want to have a look at. Some questions there around how we're influencing DECA's burning program. So you've kind of addressed that to some extent, but there's some other questions there that I can let you have a look at. So we'll now cross over to Laura Simmons. So Laura's a researcher from the Royal Botanic Gardens in Victoria, and is doing some work on the genetics of some Banksia species that she's going to present to us on today. So welcome, Laura, and thank you.
Thank you very much for the introduction, James. I'm going to launch straight into my talk, I hope. There we go. So the Conservation Genetic Group, as some background, the Conservation Genetic Group at the Royal Botanic Gardens Victoria works within the Science Division to provide genetic information that aims to maximise the long-term success and resilience of conservation and restoration programs for threatened flora. So we provide information to assist with the collection of material to maximise the genetic diversity held in ex situ conservation collections, such as the Victorian Seedbank, as well as living collections that are held at the Botanic Gardens. We also aim to identify natural populations that may be in need of genetic rescue to enhance diversity, and determine where to source propagation material from, with the aim of alleviating things like inbreeding depression and aiming to boost evolutionary potential. And therefore our work informs translocations. Occasionally, our work ventures into the species delimitation space, and can also therefore help with taxonomy. Over the last 3.5 years, Tara and I have been working on the conservation genetics of approximately 35 threatened flora species in Victoria, and today I'll be talking about 2 of the Banksias. So the first is the hairpin Banksia, which doesn't need much of an introduction after what you've just heard from Annette. However, I thought I would highlight that the hairpin Banksia species complex has a wide distribution across the east coast of Australia, and there is a complicated taxonomic history. And this is shown here by the different colours across the distribution in the map. And up until late last week, Friday afternoon at 4 p.m, I believe, there were inconsistencies in the taxonomy between the states. There's a high level of morphological variation that exists across the species complex range, and this was quantified by Stimpson et al. In 2016. However, a recent genomic study by Wilson et al. That Annette mentioned tested the genetic structuring between the geographically localized morphological entities that were identified by Stimpson. And this genomic work found that there are 2 major clades within the hairpin Banksia, confirming that Banksia cunninghamii and Banksia spinulosa should be recognised as separate species. Within these clades, genetic variation is morphologically cryptic, clinal in some parts of the distribution, and forms discrete metapopulations in other parts. So this diagram here shows the cunninghamii clade, and within it, genomic diversity is partitioned by the Southern Transition Zone, the STZ on the map. And our genetic work focused on the southern part of the cunninghamii clade, in particular the range where plants have no lignotuber and therefore rely on seedlings post-fire, as this is a major life history trait that influences how genetic diversity is maintained in the landscape. So to answer our questions around genetic diversity and structure in the southern group of Banksia cunninghamii, we collected samples from 24 locations across Victoria. This included the area in East Gippsland that was affected by the Black Summer fires. Here, seedlings were sampled, and then adults were sampled at all other locations, which included Cahoon State Forest, Southern Foothills, Strzelecki Ranges, Wilson's Promontory, French Island, and the Dandenong Yarra and King Lake ranges, which Annette collected for our use. At each of these locations, we collected leaf material from 5 to 10 individuals, which was then sent to Diversity Arrays Technology for DNA isolation and whole genome reduced representation sequencing, DART-seq, and we analysed the subsequent SNP dataset. So our results found that all populations were inbred. This is indicative of small fragmented populations where mating is occurring between closely related individuals. Gene flow is also restricted between populations, and this is likely due to the limited seed dispersal capacity. I just want to highlight some of the abnormalities here. The population in Cahoon State Forest has very high inbreeding coefficient and a very low genetic diversity values. And this kind of highlights the isolation from other populations, but could also potentially hint at things like clonality happening in this population. We identified 2 geographically defined genetic clusters with a strong split between locations in East Gippsland and locations across the remaining distribution, the Dandenong Gippsland group. At the bottom is a principal component analysis. Each dot represents an individual with the colours representing populations. The PCA shows strong genetic structuring across the horizontal axis. And then the figure on the left is a structure plot with each bar representing one individual grouped by population and the assignment to 2 significant genetic clusters. So of interest is that while being geographically closer to the East Gippsland group, the Cahoon State Forest location is genetically more similar to the Dandenong Gippsland group, and in particular a population from the Upper Yarra area. So our conservation recommendations for this species is that for ex situ conservation, such as seed banking, it's important to conserve genetic diversity from both of the main genetic clusters. That's the East Gippsland group and the Dandenong Gippsland group. And while I didn't go into detail for each group, for restoration purposes, it's important to know that there was additional genetic structuring within these two broader groups. And therefore, it's ideal to use material from the closest geographic possible areas that are possible. For in situ conservation, it's important to ensure that fire intervals are between 10 to 20 years to maintain the genetic diversity of populations. This allows time for the replacement of adult plants that are killed by fire and allows for several seasons of cones to develop. I guess just remembering that each flowering season is an opportunity for genes to move. And something that I just wanted to leave here as a thought that came up in a discussion with Tara on Friday is that potentially the downside of highly detailed mosaic burning is that if you have neighbouring populations at different age classes or stages, such as one being an adult and one being juvenile, there will be an asynchrony develop in terms of mating and therefore potentially less opportunity for gene flow, and leaving the only available mates as those that are nearby, which could be closely related. So I guess any decisions need to be made kind of on a very local basis. Shifting on to the other Banksia we've been working on, this is the Mountain Banksia, Banksia canii. This is a spreading shrub to 3 metres tall that occurs on sporadic ridges in subalpine New South Wales and Victoria. This species is listed as critically endangered under the FFG Act, and like Banksia cunninghamii, it is a serotinous obligate seeder. Much of the distribution was affected by the Black Summer bushfires, shown in the shading on the map. And while there's been significant seedling recruitment in some parts of the species range, and the photos here of seedlings in the middle, there was one population, the population at Mount Burrawa in northeast Victoria, that is now locally extinct. No seedlings have been observed post-fire. So again, our genetic questions were, what is the genetic diversity across the range and how is this structured in the landscape? And then if we're going to restore the Mount Burrawa population, where do we source the material from? To answer our questions, we collected samples from 15 locations across the range. We included herbarium samples from the Mount Burrawa population, and these were collected in 2017, so fairly recent. Following the same methods as previous, we used a DartSeq SNP dataset and analysed this. So having a look at the results, observed genetic diversity was fairly consistent across most of the populations. All populations showed levels of inbreeding, with again low gene flow detected between populations. This again highlights that localised mating is happening between closely related individuals, and it's consistent with the isolation of populations and limited seed dispersal capacity. But one thing I'll point out is that genetic diversity was particularly low in the eastern distribution, and inbreeding coefficients were particularly high. So like the Cahoon State Forest population of the hairpin Banksia, this could indicate that there's some level of clonality occurring in this part of the distribution. In terms of genetic structure, the PCA plot at the top there is showing strong genetic structuring, particularly across the horizontal axis. The structure analysis identified 4 significant genetic groups, with the bar chart showing individuals in populations and their respective assignment to 4 groups. So when we change this structure chart to a pie chart for each population plotted on a map, you can see quite obviously that the genetic groups are geographically based. And this pattern is consistent between the PCA and the structure analysis. So to take you from here on a small tangent into the realm of morphology and taxonomy, with a caveat, I will return to genetic results in a moment. That the interesting thing here about these 4 genetic groups is that they align to 4 geographically defined morphological groups in Banksia cani that were termed topodemes by Alf Salkin in the 1970s. If you've never heard the term topodeme before, I can probably just about say that you'll never hear it again. It's a very unique term. Used only for this species. The morphological differences in the topodemes are demonstrated by the variation in the leaves in this image. Interestingly, Salkin did a glasshouse experimental study and he grew seedlings from each of these topodemes. The plants retained their form when grown under the same glasshouse conditions, highlighting that the morphological characteristics are under genetic control rather than a phenotypic response to the environment. So fast forward 4 decades, and over the summer just gone, Andre Messina and I were lucky to have a fantastic Willis student, Francis Rowlands, based at RBGV. Francis worked on quantifying the morphological features in the Mountain and Banksia from across the range, and this included looking at things like form, leaf serrations, leaf length and density of leaves. Now, I'm not going to give too much away as this is truly a work in progress, but there is strong support for the 4 morphological groups. And based on the combination of genetic and morphological results, we will just describe 4 subspecies. And I've never considered myself a splitter. However, doing things like describing subspecies within a broader species allows us to break down species into conservation units that can be accurately assessed against IUCN criteria. So shifting back to the genetic results, our final question was how to restore the Burrawa pine mountain population. So our herbarium samples from the Burrawa population clustered with the snowy mountain samples, that is populations from Kosciuszko National Park. Therefore, maximum diversity could be created in a restored Mount Burrower population by sourcing seeds from multiple individuals from multiple populations in the Snowy Mountains genetic cluster. However, the problem here is that due to the Black Summer bushfires, only one location is composed of adult individuals, and there is therefore only one available seed source. Now, a solution might be that there is a new population recorded in the Kiewa Valley, but we haven't analysed this population. So I guess these results highlight the importance of genetic testing prior to restoration efforts, as in this case, we have identified that there's significant genetic clusters, and using plants from one group, say from Omeo over the Snowy Mountains group, might result in the introduction of plants not suited to the local area. But I guess this also highlights some of the complexity in restoration in the era of climate change. What option is going to be the best climatically suited for future climates? So for now, together with Annette and my colleague Glenn Johnson, we have several proposals in for our ideal scenario to restore the Mount Burrower population of Banksia cani. This work will include sourcing seed from genetically and morphologically matched populations. Now, with the evidence here, we know that there is one Kosciuszko National Park location that's a good match for genetics, morphology, and seed availability. However, further genetic work is needed to validate whether the Kiewa Valley population is a good option. We're also hoping to crack some of the germination requirements. Banksia cani requires a heat treatment to open up follicles on the cones and release the seed. Then the seed requires a cold stratification before germinating. And while laboratory-based germination testing will be useful, it's also quite appropriate to do nursery-based testing, and we've got a fantastic community nursery in Wodonga on board to help us out with this. As ultimately, it's that nursery that will be working to produce approximately 500 plants to go back into the ground across 2 locations on Mount Burrower. So part 2 of the ideal scenario for restoring the Mount Burrower population is to incorporate a greater ecological understanding of the species by looking at the response to fire. So we're fortunate that with Glenn's connections with forest fire management, and thanks to help from the Hume NEP team, prior to a planned burn in late April this year, we were able to set up some fire exclusion plots within areas that had different fire histories. And we made some measurements pre-fire. So this will allow us to monitor the response to fire, including seedling emergence and the time to reproductive maturity at this location. And I guess overall, this opens up opportunities to expand the ecological knowledge of different subspecies in Banksia cani and provide information to feed into models such as those that were created for Banksia cunninghami, and for use in determining the appropriate fire regime that's required to maintain populations, as well as for use in the population trajectory modelling, which form part of the assessments for listing species under the IUCN Red List Criteria and FFG Act. So finally, my take-home points are that both Banksia species had landscape scale genetic structuring, and it's important to capture this in our ex situ conservation. Also, that resolving taxonomy is important for identifying meaningful units for conservation, and then meaningful conservation, particularly that happens on ground, is the result of combined ecology, morphometrics, taxonomy, genetics, and horticulture. So it's quite a holistic art. And finally, just some acknowledgements. There's been a huge number of people that have been involved in this project and have listened to me talk about both Banksias a lot. But I'd particularly like to thank the RBGV science and horticulture teams, the agencies who've helped out with collections, Glenn Johnson, and also funding has come from the DECA Bushfire Biodiversity Response and Recovery Program, Cybec Foundation, and the Wetton Environmental Trust. Thank you.
That's great, Laura. Thank you very much. A fantastic presentation and some amazing work there to understand more about the genetics of different populations of those 2 species. We've got Andy's going to organise the questions from the chat, but maybe before we go there, maybe for both of you. So for the species that you've touched on today, you've both mentioned, the sort of conservation actions that might be required to help ensure the persistence of those species going forward. Could you talk to those in some sort of summary, what you think those actions are, and where might be perhaps the uncertainty or where we need to get further knowledge to inform some of those actions going forward? Who wants to have a crack at that one first?
Well, perhaps I'll start with the fire management question. I know there's been quite a few questions in the chat about that too. Yeah, it's a difficult one. It's a big challenge, and I'm sure there'd be answers from a lot of regional colleagues who are listening online too. But I suppose what I'd say with this is that the fire intervals that we've looked at the fire intervals that came, the results on fire intervals that came out of research, those thresholds at individual sites can be more or less. So I think it really points to the fact that we really need to check on ground what's happening in a burn unit. Are there plants with cones? Is there seed source available for them to reproduce? Is the time of year suitable? Are we in a drought? Let's not burn because the seedlings won't come up. Is there the possibility in those zonings where you don't have to burn the whole unit, where you can exclude some patches of Banksia if needed? All of these things are being looked at, I know, by regional fire planners. Obviously, there are limitations in those areas very close to houses, but in the other zones, I know there is that greater flexibility. So I think it really points to the fact that the modelling and our results give us the overall picture, but there's nothing beats really looking at what's on the ground. So I suppose that's from the fire planning. That's a very incomplete view of, fire planners are very complicated and there'll be different ideas about that. But I think the other thing is informally it's being treated as a threatened species by people in DECA with their planning. We need to kind of make that happen, I think. So I think that's probably enough from me on that side of things. There are many other actions, obviously, that to do with its conservation apart from fire management. And so, yeah, over to you, Laura, with that part of it.
So I think kind of first and foremost, the ex situ conservation collections that we have currently don't represent the kind of the genetic diversity that we've detected in these Banksias. So I think we've got, and my colleagues at VCS will probably contact me after this and make sure that I've said the right thing, but I believe we've only got one of the two genetic clusters in Banksia cunninghamii represented, and then one of the four in Banksia canii represented in our seed collections. We have an additional seed collection for Banksia canii that's currently being cleaned, but that's also the one that we need to do genetic testing on to figure out which morphological group that it belongs to, because right now it's morphologically looking a little bit borderline, and so genetically it'd be nice to figure that out. And then I can provide the long list of all of the research that I would like to do on these Banksias, but I potentially have summarised some of that in the part that I was talking about, the restoring the burrower population. So just some additional genetic work to try to figure out where some of the boundaries are, and yeah, working on that taxonomy as well.
Thanks, Laura. And there's some interesting comments in the chat about what's ultimately the aim for restoration. Should it be about maintaining local provenance versus, essentially gene mixing to try and, amongst other things, help species adapt as best they can through climate change? It's probably not a science question, but is there a sense, what do geneticists have to, do they have any views on that particular problem?
That it's very complex problem. I think something that I would be really interested in doing in future genetic work is doing some of the genetic modelling of what would happen if we either combined some of our genetic groups or kept them sort of as a single entity, and look at how that changes over time, whether genetic diversity can be maintained or whether it starts to peter off without having the influence. I guess, as always, we would generally err on the side of trying to capture as much genetic diversity as you can from within a like a genetic group. And
could I add to that, Laura, of course, and just highlight the work that's being done by the Friends of the Forgotten Woodlands on silver banksia in Western Victoria, and also the Euroa Arboretum. That's been a great collaboration, hasn't it, between universities and, you know, government agencies, private landholders, and I think there is that experimentation going on, isn't there, with either genetic mixing or keeping seeds apart in their seed orchards. So that's something that Steve Sinclair from ARI is involved in as well, and I think there are some links through the Swift Banksia page, so people could follow up on some of the journal articles and stuff about that. Yeah.
Thanks, Annette. Thanks, Laura. It is a challenging space. So with that, I might hand over to Andy and Tess. So you guys have been going through the chat. Do you want to perhaps come back to some of the more pertinent or most popular questions that people have put into the chat?
We sure can, and keep them coming. We will do our best to get through as many as possible, but also we share them with our presenters so they kind of have an idea of the questions that are out there in the mind of the broader ARI seminar community. So thank you very much for them. I'm going to throw to Annette with this one, and this is about the future of conservation for hairpin Banksia. You talked a lot about the fire threats, but I'm sure there's interactions with other things.
Sure.
With weeds, maybe deer or herbivory or pollinators or even Phytophthora. Have you done much work about those interacting threats?
Yeah, it's interesting because, and I'd like Laura to jump in here on the pollination side of things, because there is that question if you've got these kind of isolated populations, other pollinators, you know, what's happening with the pollinators. So I guess I was involved with a student at La Trobe Uni doing a small project on that, but there's a lot more that could be done in that space, because there is a real variability about this. Some populations produce a lot of cones and seed and others don't, and you know, what's going on there? Is there I'm way outside my area of expertise, but is it a genetic issue? Is it a pollinator issue? And I think there's, a lot of work to be done on that. Laura, can you add to that side of it?
I think you've summed it up pretty well.
Well, that's a wonderful answer, Annette. Thank you. I think you did sum up very well. In that case, we'll send Laura a different question. A lot of people are asking about like the genetic mixing of maybe garden varieties of Banksias and wild populations, or if different sort of crosses could be more suitable for the future, I guess, with climate change. Do you have any perspectives on that?
Again, it can be such a philosophical question. Yeah, I think it's quite interesting to have a look if you can match things like, you know, any of the traits to genetics and climate variables, then potentially we could do a lot of selecting for, you know, appropriate scenarios under different climatic conditions. We don't have that level of detail and data from our work. In terms of nursery plants versus wild populations and crossing, I think there's a couple of things there that nursery plants, it can be really hard to determine what the provenance is and what the background is. In a lot of cases that I know of, sometimes seed may be collected from one or two individuals, that's grown into a mature plant, and then seed is then harvested from one or two, or cuttings are taken from one or two plants in the field. And so what you have represented there is just a small segment of the genetic diversity that's in the wild. So I guess when we're talking about wild populations, we like to still err on the, I guess, the precautionary aspect of matching what was there with, with local stuff.
Great, thank you. It sounds, again, more complicated.
And I should highlight here that we didn't actually analyse any of the nursery stock, because, Banksia spinulosa is probably, it's very, very widely cultivated, and there are so many different cultivars known, and a huge can of worms there in particular.
An important caveat then.
Yeah.
This question, I guess, goes both to both your talks, and that you really pushed that 12-year fire interval as a minimum to help things survive. And then Laura, you spoke about the local extinctions of some species. Lucas had a really great question about what does the sort of fire sensitivities and this life history of Banksia tell us about bushfires in the past, pre-colonization? Do you guys have any thoughts on that? He said it's a bit of a starter question. I think it is, but I think it's a nice one to sort of think about. I'll throw to you, Annette, first, even though I can see your thinking brain is on.
Look at my face going, oh my goodness, that's a curly one.
A hairy one, a hairy pin one.
A hairy. Well, I suppose. Well, it tells us if they take a certain amount of time to produce seed, that they can only tolerate, fires at certain, that sort of interval or longer. What else could I add to that? Yes, I suppose the interval question is reasonably straightforward in that has a bit of a window in the past. So they're fairly long lived. They still, you've got plants that are 30 or more years old still producing seeds. So, and there is occasionally some interfire recruitment where there's just the cones will open after a few years. In a warm summer, the cones will open. If there happens to be a bit of bare soil, there'll be a bit of germination, a few plants. So I wonder whether in the past there might have been more multi-aged stands. So that's a guess, but I suppose that's feasible. That if there was a great mosaic of patchiness of burns across the landscape, then that's conceivable. And whether perhaps in the past the landscape being more open, more small mammals digging up all the things that dig up the ground, and more bare soil for the seeds to hit and actually germinate. So I think you could sort of speculate about those things kind of reasonably. Yeah.
I think it's a great answer. Laura, did you have any thoughts to add?
Yeah, I guess I will go on a tangent about the Mount Burrower population after I try to address the question. But I guess it really is still quite an understudied kind of area, particularly in like temperate forests. So I guess we know that the combined effects of droughts, fire severity, fire frequency, and plant growth characteristics do influence their ability to, I guess, persist in a fire-prone environment. I guess things like having a lignotubor is a characteristic that enables the species that may have low fecundity or low recruitment rates to persist where site conditions are, not conducive to seedling recruitment, and I guess persist in areas that do have very high fire frequencies. Yes, shorter fire intervals can be detrimental with seed production. We know that, and seedling establishment, and I guess that can lead to reliance on vegetative reproduction over time. So there's kind of a little bit of thought about whether plants that have lignotubers are an evolutionarily older kind of thing, because they have had to survive in a lot of different conditions, whereas things that still seed are just happy in the current conditions. The only thing I was going to mention, I guess, about the local extinction at Mount Burrower is that there was kind of a real combination of things that happened there. From what I can piece together from mostly speaking to the rangers, because I wasn't in the area at that time, there had been an absence of fire for over 50 years in those populations. The plants were quite large, very, very large for the species in terms of trunk girth, and there hadn't been any fruit set kind of observed in those populations for quite some years. And any fruit that had been set was getting predated by black cockatoos. So there's kind of a combination there of something happening with the ecology and cockatoos doing a bit of predation, maybe a bit too much predation, and also plants that have gone to a reproductive state and reached peak adult maturity and are now going through a senescent state and are therefore not reproducing in the population. So just another interesting factor about the species.
Yeah, that's incredible. Thank you for that detail. Really appreciate it.
Thanks, Laura, and thanks, Annette, again, for some really couple of fascinating presentations. I'd like everyone just to put their hands together, virtually or otherwise.
Look, there are a few other questions there, James, that we didn't get to, obviously. I just wonder, people could email in or something. There was Phytophthora that's concerning. What else did I see? Anyway, that was one that caught my eye. Yeah.
No, so if there are questions in the chat, noting we couldn't get to all of them, but if there's outstanding questions there, feel free to email Annette and Laura directly. Of course, as is the often the way with these seminars, the science reveals lots more questions about what it all means, particularly in a changing environment that we're dealing with. So the past is not always a great guide for the future, given climate change and the like. So it's a complex operating environment. So thanks again, everyone, for attending. We had over 160 odd attendees, I think, so that's fantastic. The next seminar will be on Monday, the 15th of July, and that'll be about protecting birds in a changing environment. So again, highly relevant to sort of complex environmental management and decision making. Please make sure you're signed up to the seminar mailing list. I think Tess will put those in the chat, and also please sign up for the ARI updates and seminars, and you can provide some feedback as well on the seminars as well. We're always looking to try and improve these to make them as effective and as useful as possible to a range of audiences. So keen to get your feedback as well. So thank you again for attending. Annette, Laura, thank you again. Some really fascinating presentations, very thought-provoking, and more work to be done. So thanks all. See ya.